Hey, welcome back to what is ostensibly the last lecture. Yay! So, in this last lecture, we're going to talk about animal development. And some of the hallmark features of this is that while there clearly are a lot of differences in the development of a variety of animals and plants, there are some key features within animals that are the same. Um, so we're going to cover the major highlights of development, but bear in mind that we're barely scraping the surface. Development is a fascinating topic of study, and I highly encourage you to take a class in development to fully appreciate all of the different aspects of it. In your reading guide, by the way, so under Learning Objective 1, it's the first learning objective is to describe the major events that occur during human gestation. And you should be able to answer all of those questions very easily with those pages in your textbook. So I'm not going to cover that material. I'm going to leave it to you to cover. And we're going to pick up with the next learning objective, describing the genetic control of animal development. Now, in order to, no idea why that's popping up. In order to understand development in general and the genetic development, the genetic control of development, you need to have some key concepts under your belt. So the first is stem cells. So stem cells are defined here as being an undifferentiated cell of a multicellular organism that can give rise to indefinitely more cells of the same type, and from which certain other kinds of cells arise by differentiation. So in other words, a stem cell is one that can produce more of itself, and then each of these cells can then later differentiate into a number of different types of cells. So skin cells, muscle cells, neurons, blood cells, whatever kind of cell you need in the body. So it's from these initial stem cells that derive from the union of sperm and egg that then all the other cells in your body become possible. Okay? Okay. So once you actually have the union of egg and sperm, you are then going to have a series of mitotic cell divisions that are going to first form into stem cells. And there's going to be about 5 days of development, depending on which species you're talking about. But in general, it's a few days of development. And then after that point, those stem cells start to differentiate into all the different kinds of cells that are in your body. But it takes a period of time. So all of those initial cells that are developed in those first 5 days are embryonic stem cells. So they're undifferentiated. After that point, the cells begin to differentiate, and that differentiation, so becoming a particular type of cell, so say a pancreas cell or a skin cell or a muscle cell, a nerve cell, so differentiating into those different types is controlled by genetic mechanisms. And in other words, genes are turned on and are producing products, or they aren't. And what we're trying to highlight in this figure here is to show you 3 different kinds of cells. And it's possible for there to be genes that are turned on in all of those cells at some particular stage. Some are going to be on while others are going to be off. And certain kinds of genes may be completely off. It depends on what that cell is. So differentiation is determined by the differential gene expression that's going on in that cell. Now, gene expression is controlled a number of different ways. It can be controlled by transcriptional control, so something that's regulating transcription being turned on or off. You can also change gene expression by alternative splicing of mRNAs into and out of segments of the strand. There can be selective destruction of mRNAs. You can change the rate of translation. And then the activation or deactivation of certain proteins will control expression of genes. So the most important of these to development is transcriptional control by a long shot. It's not to say that these other processes aren't involved, but transcriptional control is far and away the most important in development, and particularly in early development. Now, before we get too far down the road, though, so as those cells begin dividing in those early stages and they're undifferentiated, um, you will eventually get to this point where they will begin to differentiate. And so there is a transcriptional control or a genetic mechanism for determining what that cell is going to differentiate into. But a really important aspect of what determines the fate of the cell is where it's located relative to the embryo. So where it's located in terms of the body axes. And what's also important is a time element. So depending on the stage of the embryo, wherever that cell is relative to the embryo's axes, so anterior, posterior, dorsal, ventral, left and right, depending on where it is relative to that, and then the period of time or the stage is going to determine what the fate of that cell is ultimately going to be. And that's heavily influenced by all the other cells around it at that time. And let me show you some other slides to kind of explain this. I have to admit that a lot of this is very, very simplified, and to really fully understand development, it's really important, I think, to take some classes in development. But I'll try to explain it as best I can here. So there are these morphogens. So these are molecules that are in the embryos that are pattern formation molecules. And so they impact what happens to cells on a concentration gradient. So there are certain kinds of cells that when morphogens are at some sort of threshold level of concentration, it activates genes in those cells to perform certain functions, and this changes what the cell may turn into. And so what this graph is trying to show you is that if you have morphogens being plotted on here, here's the concentration of the morphogen. So different cells have different thresholds at which they are activated on by these morphogens, and then will begin to differentiate into different cell types. So one of the most famous examples of really important morphogens early in development is bicoid. So bicoid is an mRNA that is made by cells from the mother, and then they're transferred into the egg. And ultimately, this will cause the development or the production of bicoid protein. And so what you're seeing here is a scan of an embryo, and it's been stained for the presence of bicoid mRNA. And what you should be taking away from this picture is that, so this blue, so it shows that there is some bicoid all throughout the embryo, but it's most prevalent in the anterior end, which is why you get the red. So the closer to red you are, the higher the concentration. So the lower down on the scale you are, the lower the concentration. So yes, you can see bicoid mRNA kind of sprinkled throughout, but it's in a very definite concentration gradient where it's heavily concentrated here in the anterior end, and it's kind of decreasingly present as you extend towards the posterior end of this embryo. And similarly, if you stain for the presence of bicoid protein, so the product, then you find also that's highly concentrated here in the anterior end. You have some bicoid protein present around the margins, but not so much in the center. So in this case, I should probably show you this slide. So bicoid is a regulatory transcription factor, so it turns transcription on. And clearly what's happening is in this particular case is it's turning on transcription into high mode, if you will, on the anterior end of the embryo. But then you're finding that it's in lower and lower concentrations as you move away from the sort of nose or anterior end of the embryo. So you still find it throughout the embryo, but just in lower and lower concentration. So there's a whole number of morphogens that influence cells in this way, will influence the turning on of transcription. Pardon me, oak pollen's still getting to me. So these kinds of morphogens are going to be present throughout the embryo and are going to influence their differentiation into different types of cells. And it's a dynamic process that's happening throughout the embryo in time and in space. And so all kinds of other morphogens are turning transcription on throughout the embryo as it develops. And so this is a dynamic process that's going to continue until until the baby is born. So these regulatory genes, much like bicoid, are often found in cascades or in genetic regulatory cascades. And so these regulatory cascades are defined as being when one gene gets turned on, it will then influence another set of genes to get turned on, and then they will influence another set of genes to be turned on, and so forth. And so what we're showing you here are some really important cascades in fly embryos, although to be honest, these are also present in a lot of animals, not just flies. It's just that they've been better studied in flies. But you find morphogens, which establish a broad anterior-posterior region of the embryo. So that's one of the first things that has to happen. So once we get past, once we get to gastrulation, and I'll show you this in a minute, what's going to happen to the embryo is you're first going to establish an anterior-posterior axis. Then that's going to turn on gap genes, which is going to organize the cells in the embryo into groups of segments along that anterior-posterior axis. So it's going to be segments, different segments along the embryo. Those gap genes are then, once they start production, they're going to turn on the pair rule genes, which is going to organize the cells within each segment, so within each of the segments of the embryo. Those genes are then going to turn on the segment polarity genes, which are going to establish anterior-posterior gradients within each segment of the cell of the embryo. And then Hox genes can get turned on. That's going to trigger the development of other structures, so things like antennae and wings and limbs in the case of a fly, or it's going to have an influence on limbs of all animals. T-box genes are also going to get turned on. And then those genes are going to impact, are going to turn on effector genes, which are going to change the proliferation of cells in certain areas. So like maybe to form your fingers, and then it's going to ultimately affect apoptosis, which is going to reduce the webs between your fingers so that you actually have fingers. It's going to influence cell death, movement, and differentiation of cells. Okay, with that really heavy backdrop in place, let's talk about the events of cleavage during animal development. And what you're seeing here is cleavage happening. So this is a great video that begins with the initial single cell and then goes through the different stages of cleavage until you reach gastrulation. Wheelie, 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 just spins around once it turns into a gastrula. Okay. So if we use human development as the sort of backdrop for this discussion, we've already covered, the union of egg and sperm to form the fertilized egg. And it's after this point that you have several cell divisions that are referred to as cleavage. So you're dividing the DNA as well as the contents in the cell. And there's going to be several stages of cleavage that will ultimately form what's called a blastula. And we'll talk about the details. After the blastula forms, it's going to transform its shape into what's called a gastrula, and I'll explain this in some detail. And after this point, you're then going to have the development, or you're going to go into a stage called organogenesis. So all of the organs in the embryo will develop. And I know this looks quite bizarre here, but this is an early first trimester embryo. And at this stage, pretty much or by the end of the first trimester, all of your organs will have been formed. And then the next two trimesters basically consist of growth. A little bit of rearrangement, but basically it's growth until you finally reach birth. So let's go through each of these stages in a little bit more detail. And bear in mind that these major stages are the same throughout pretty much all animals. There are differences between species, and in some cases those differences can be dramatic, but the basic things that I'm going to tell you are the same throughout animals. Okay, so cleavage consists of a series of rapid cell divisions. And when these cell divisions happen, notice in this figure, here's the fertilized egg, and then here's the blastula, which is basically a big hollow ball of cells. And what's really important to take home from this is actually shown in relative size. So the blastula is about the same size as that initial fertilized egg. The difference is that with each of these cell divisions, the cells become smaller and smaller and smaller while maintaining the overall size of the embryo at that stage. Now, after this stage, it's going to change in size dramatically, clearly. But I want you to take home the point that with each cell division, you get more cells, but each cell is smaller. So each of the cells that is produced during cleavage are called blastomeres. And the mass of blastomeres is called the blastula or the blastocyst in mammals. And that's what you're seeing here. So it goes through a number of cell divisions. And by about day 6, what you have is, so this is the blastula. These outer cells here are referred to as the trophoblast. And it's really just a hollow ball of cells. So you can think of it as being like a soccer ball. And the inside of this hollow area is referred to as the blastocele. But there's a second population of cells called the inner mass that you see right here. And so what's going to happen is the embryo itself is going to develop from this inner cell mass. The trophoblast is what's going to form the placenta. And the placenta is what's going to form a very important structure that is going to implant. It's going to develop at the point of implantation, so where the zygote is actually going to implant in the uterus. It forms a hugely complicated capillary bed in the uterus, and then it's going to make the connection between the mom and the embryo. Okay, so through that connection, of course, is going to be the umbilical cord. So what that's going to allow to happen is for there to be the exchange of nutrients and gases and the removal of waste between mother and developing baby. But do bear in mind that what's not going to happen is there's not going to be a transfer of blood between the embryo and the mother. Just the transfer of nutrients, gases, and waste products. So I should probably point out that the blastocele is a fluid-filled cavity, so it's not just open space. There is fluid in there. Okay. So now let's talk about those different stages. So we got up to blastula. What happens after that? Well, what happens after that is gastrulation, and you're seeing an absolutely amazing scanning micrograph of this happening. But let me explain this in a bit more detail. So there was a famous developmental biologist, Lewis Wolpert, that said it is not birth, marriage, or death, but gastrulation, which is truly the most important time in your life. And it truly is. Like, it's hard to relate to maybe at this moment, but this is a stage at which everything begins to differentiate and happen. So you're going to have the development of these body axes. You're going to have the differentiation of cells into different tissue layers, which are then ultimately going to determine the fates of all of the cells downstream from there. So if something goes wrong in gastrulation, everything is going to go wrong with the embryo. So here is the blastula, stylistically represented. And gastrulation is this event where you have this what's called an invagination of the blastula. You see diagrams like this all the time being represented, and I wish I had a balloon on me, but I don't. But I'm going to use my porg because it's the closest thing I got to a balloon. And think of gastrulation. So imagine that this is not a porg, but it's actually a balloon. If this were your blastula, imagine if you could take your finger and press into a balloon and kind of poke it into the balloon so that your finger was like most of the way through the balloon itself. That basically represents what's happening in gastrulation. So you have this pooching in of the ball of cells, and what's going to happen is where this invagination happens, ultimately it's going to make this big opening in the ball of cells, and it's actually going to end up going all the way through to the other side, which I'll show you in just a moment. So when this happens, you have then the formation of two or three, depending on what animals you're talking about, 2 or 3 very important tissue layers. So you're going to have the development of the ectoderm on the outside, the endoderm on the inside, and then for those animals that have it, the mesoderm, which is going to be this middle layer. Now, I'm not showing you that here in this slide. That's going to come up next. So here we define this process. So you have extensive and highly organized cell movements and changes in cell shape that rearrange the embryonic cells, or they're still stem cells at this point, and they're going to then rearrange into the gastrula. So, depending on, so I don't know how in depth you went into this in 111, but there are different kinds. The animals have been categorized into two different groups, the diploblast and the triploblast, on the basis of the number of embryonic tissue layers that form in the gastrula. So you either have ectoderm and endoderm only, in which case you're diploblastic, so 2 layers, or you also have the formation of mesoderm, and they're showing it just as little outpocketings here in this early stage embryo. But what's going to happen is these layers are going to develop all throughout this area in between the ectoderm and endoderm. And then you'll have 3 sort of basic embryonic tissue layers from which all of your organs and structures are going to derive. So just to show you that video again, so here's the initial egg, the initial single-celled zygote, and watch what's happening. So that cell is going to divide in 2. Each of those is going to divide in 2, so now you have 4. Now you have 8. Now you have 16. And it's going to go on and so forth until you reach a point right about here. So now you've reached the blastula stage. Now you're going to have this invagination. So you got a little cup there. And then as it forms that gastrula, it spins around quite a bit. So I'll just show that to you again one more time. Oh, I should point out that up until just before this final blastula stage, up until that point, you can remove some of the cells from the embryo, and those cells will continue to divide and form a fully functional, perfect second individual. So there is the ability to separate cells and form 2 individuals from one initial zygote, and that's sometimes what happens with identical twins. All right, let's move on. Okay. Oop, I feel like I missed one. Nope. Okay. So once you have the development of the gastrula and the establishment of ectoderm, mesoderm, and endoderm, so those initial embryonic tissues are going to result in the formation of, in the case of the ectoderm, your outer covering, so your skin and your nervous system. The mesodermal layers are going to form your muscles, your internal organs, not all of them, but most of them, and your connective tissues. And the endoderm is going to develop into epithelial linings, like on the inside of your gut, into the liver, the pancreas, and the thyroid. So these are structures that you could commit to memory that are going to develop from each of those embryonic tissue layers. And they're indicating them to you in color as well here throughout this diagram. So remember that, so it's only after gastrulation that you, or it's at gastrulation that you then start forming those important body axes. So anterior, posterior, dorsal, ventral, and then left and right. Here we refer to what's happening with protostomes and deuterostomes because the development is a little bit different. Although I'll warn you, this is a gross oversimplification that you see in textbooks all the time. And when you look at the specifics of different animals, it's much more variable than what we're showing you right here. But, so and we're not going to go into the details of spiral cleavage versus radial, which is absolutely fascinating, but you'll have to get that later. So here's the gastrula. And there's 2 different ways that you can actually form the mesodermal layer in the middle between the ectoderm and endoderm. You can either have the development of those layers from right here at the junction between the endoderm and the ectoderm, there's a pocket of cells that form here, and then they migrate upwards and join up here. This is what happens in the protostomes. And as a result, this initial opening, the blastopore, this is going to form the mouth in protostomes, and that invagination is going to keep going all the way through until it opens up at the other end of the embryo, and this is going to ultimately form the anus. So here is your, if you will, primordial gut tube. In deuterostomes, it's a little bit different. What happens is you have, it makes it look like a Mickey Mouse head in here, but what happens is mesodermal layers develop here on either side as outpocketings of just the endoderm. And what will happen is these will grow and develop and kind of come downwards this way. And then this tube will continue all the way through. It actually ends up bending to the side. They don't show it here, but it actually doesn't go straight through. It actually bends to the side a little bit. And so now it's just the reverse of what happens in protostomes. So in deuterostomes, that first opening forms the anus, and then the mouth forms last. So I used to joke to my development class, because we're deuterostomes. So I always used to joke that you're kind of an asshole before you develop a mouth. It's not a great joke. Okay, so now that you have some of those processes in your toolbox, let's take development a little bit further and talk about what happens after that gastrulation stage and the development of the gut tube. And then, oh, I should probably say, by the way, so clearly, by virtue of forming the mouth and the anus, that's going to establish your anterior posterior axis. But how does your dorsal ventral right and left form? Well, let me show you that in this next part. So the neural tube is critical to that. So if you imagine the embryo and now look at it head on at the anterior, that's kind of what you're seeing more or less in these diagrams. And so you've got the anterior posterior portion developing, but now what needs to happen is the development of the notochord and the neural tube. So what happens is a series of cells begin sending out chemical signals, developing just to one side of what's going to be the gut, so on one part of the embryo. So as those signaling molecules kick into high gear, what it does is it makes the ectoderm, the ectoderm, shown here in blue, it makes it start to kind of pooch up. As that happens, it pooches up, forms these ridges, and basically it forms kind of a furrow along the dorsal side of the embryo. And then it the furrow forms, and then it closes together, and then forms this neural tube. So this neural tube is what's going to be ultimately your central nervous system. And this thing that they're referring to as the notochord here, it's very undeveloped at this stage, but that's ultimately going to form your vertebral column for those animals that have a vertebral column. So the establishment of this is going to help establish the dorsal ventral axis, right? And then from this stage, you can then, through a series of cell signaling stages, you will then develop your left and right orientation as well. So you have to do two of your axes first, and then the third axis comes naturally. So after this neural tube forms, you will then have the formation of somites. So these are sections of tissues that are going to form all kinds of different structures downstream. So it starts out with the neural tube, and then the somites that develop around the neural tube are going to form either the muscles of the back, the dermal layer of skin, limbs and the muscles, or it's going to build bone. And so we're showing it to you in this way, but this doesn't really highlight how these different blocks of tissue, and they're going to be paired on either side, by the way. So they're showing you one block over on one side of the neural tube, but there's going to be another block on this side, and then they're going to grow and differentiate into all of these different structures. All right, I'm going to end with that part for now, and we're going to cover a little bit more details in class. Hope you enjoyed, and I'll see you in class.